Okay, here comes to the chapter 2 of the DSE level biology revision videos. So first of all, we would like to talk about water because in our life, most of them are organic, and for the majority, it will be water. So we have to understand for the functions of waters. So here we listed 5 common functions of water. So please kindly pick like 2 of them to remember. So as a reactant, like in the photosynthesis, and also it serves as a medium of reaction, and it also serves as a medium of transport. In our body, it will be blood, and it also provides support for some organisms, and it also serves as a lubricant. And for the inorganic ions, we have the nitrite, that is the NO3 minus, and it is a source of the nitrogen and also proteins for all organisms. And for magnesium, it is a essential ions in the chlorophyll and also required in the enzyme activities. And for the iron, it is required in the formation of the hemoglobin and also for some enzyme activities. And for the calcium, you have known in form 3 that calcium is required for the strength of the bones and teeth, and is also required in the blood clotting step and also nervous systems. And for the organic matters, it is commonly known as the biomolecules, and all of them will contain the element carbon. And here are the organic chemicals. So for the carbohydrates, lipid, proteins, and also the nucleic acid. And for the carbohydrates, these 4 examples you have to bear in mind. For the glucose, it is the main energy source. The word main is an important term and you should not skip it. And for starch, it is the energy reserve for plants, and we also take in starch in our meal. And for the glycogen, it is the energy reserve in animals. That's why for human, we use glycogen. And for the cellulose, it is used to make plant cell walls. And for the lipid, there will be 2 examples. The first one will be triglycerides, and it is serve as the energy reserve, and it is in the adipose tissues to reduce the heat loss and also to protect our internal organs from damage. And this example, phospholipid, you should bear it in mind because it is required for making cell membrane, which is the chapter 3 major content. And for the protein, here are the uses of protein, and all these 4 examples you should bear it in mind. The first one is enzyme, second one is hormone, and the third one is antibodies, which is required for body defense, and the fourth one is the transport of substance like hemoglobin and also carrier proteins, which is also an important concept in chapter 3. And for the nucleic acid, we have 2 types of nucleic acid. The first one is the DNA, and then the second one is the RNA. Here are the general structures of RNA and also DNA. Based on our syllabus, you are required to understand and compare them. So with these 2 diagrams, please try to compare them. And for the DNA, it is required for carrying genetic materials, and that's why it controls activities of cell. And for the RNA, it is required for the protein synthesis. And in chapter 2, we focus on the term cells, and cells is closely related to the development of the microscope. And then we have the cell theory. Based on the theory, we believe that all organisms are made up of 1 or more cells. For human, we have a lot of cells. However, for some unicellular organisms like amoeba, they will only have 1 cell for 1 organism. And cell is the basic unit of life, and all cells will come from pre-existing cells. So don't ask me what is the first cell, okay? This is beyond our scope. And there will be 2 types of microscope. The first one is the light microscope, which is usually used in our school lab, and because the live specimen can easily be paired, and also we have colored image. And for the electron microscope, there will be 2 types. One is the TEM and another one is the SEM. And they can provide with a higher magnifications and also resolution. Remember, the term magnifications and resolutions are 2 concepts, and they will usually have the black and white images only. And there are a few components in the microscope that you should bear in mind. There will be 2 sets of lens. One is the eyepiece, another one is the objective. These 2 terms you should remember them, and based on the term, you will know that this is the lens close to your eyes, while this lens is close to your sample. And we will also have condenser and diagram. However, in the modern type of light microscope, these 2 may not be easily found. However, for the adjustment knot, there will be always cross adjustment and also fine adjustment knot. And your sample will be placed on the stage. And remember, for the properties of the image, it will be laterally inverted and also inverted. So remember, this example will help you in the exam. And here are the procedure to use the microscope, and I highlighted the two most important terms. That is, the first one is always use the low power magnification first, and also to lift up the stage to the maximum and then go downward. This will be the general important procedure when using microscopes. And in order to compare the low power and high power images, for the high power one, we will focus on a smaller area with more details. However, the image will be dimmer. And in order to calculate the total magnifications, here is the formula. So you, with the eyepiece magnifications, multiply with the objective one. And the X is the unit, which is like the 5 cm, the cm. So don't skip it. And this is how we may convert the size of the image to the actual size. So remember, in your exam, please bring a ruler with proper marking. And as we are converting different units, here are the general table for the conversion. And you should notice that cm is not commonly used because it is not a basic scientific investigation unit. And here is an example that you may try to apply your skills. And for the structure of the cells, all cells should contain cytoplasm and cell membrane. However, we should be able to compare things based on our curriculum. So the first one we are comparing with will be animal and plant cell. For the plant cell, it will definitely have cell wall, and for the plant cell, they may have chloroplast. Remember, it is may, so it is not a must. And also for the plant cell, they usually have a large central vacuole, and it is usually not a must. Okay, so for the plant cell, it will usually be larger and regular inside. So you may notice that from point 2, 3, and 4, they are not a must, but for the point 1, it is a must. That's why in the exam, always remember the first rule. And we will talk about the organelles in the cell. The term organelles means organ inside the cell. And the first one will be the nucleus. It's bounded with the nuclear membrane and it contains the DNA. That's why it controls the activities of the cell. And for the cytoplasm, it is the site of many chemical reactions, and it also allows movement and transport of materials inside the cells. And for the transport and movement within and across the cell, we will talk about it in chapter 3. And for the cell membrane, it's important to notice that it is composed of the phospholipid and also proteins, and it is used to separate the cell contents from the outside. And these properties, differentially permeable, is a very important term, and you should always state it when you are answering related questions. And that's because of the differential permeability, it controls the movement of substance across the membrane. And for the cell wall, it is thick and rigid, and it is made up of the cellulose, which is previously discussed, and it is fully permeable, which is different from the cell membrane. So with its rigidity, it helps to protect, support, and give the shape to the cell. And remember, for the cell wall, it's only present in the plant cells. And for this term ER, the full term is called the endoplasmic reticulum. There will be two types. One type is the rough ER, and other one is the smooth ER. And for the rough ER, is responsible for the protein synthesis, and for the smooth ER, is responsible for the lipid synthesis. And the difference is whether it has or not, with or without the ribosome. And that's why ribosome is involved in the protein synthesis. And for the mitochondria, for the plural term is called mitochondria, is a double membrane structures, and it is a site of respiration and also for the energy generation. And for the chloroplast, it is also double membrane, and it is present in all green plant cells, and it contains the chlorophyll. Remember, the term chloroplast is always larger than the chlorophyll because chloroplast is an organelle, while chlorophyll is a pigment inside the chloroplast. And you may also notice starch granule inside. For the vacuole, it is present in both animal and plant cell, but usually in animal cell it is smaller and fewer, and it is single membrane, and it contains water, enzyme, and other things, and it is required in the support in the plant cell. So here is a schematic diagram of how we should draw a cell. So always remember to bring an HP pencil with you and use clear labeling and a proper title. And straight lines should always be used in the labeling. And for the lines included, it should be smooth and continuous without looking like any drawing in your visual art lesson. And we will also like to compare two types of cells. Previously, we compared animal cell and plant cell. Now we are comparing prokaryotic cell and eukaryotic cell. For the prokaryotic ones, their DNA is lying freely in the corpus, which means that it doesn't have a true nucleus. And all of them will be unicellular. The best example will be E. coli. And for the eukaryotics, the term "eu" means true, and it has a true and defined nucleus. And we human is an example, and also amoeba will be another example. And we should be able to compare them. For similarities, they both have cell membranes and DNA. However, for prokaryotic cell, it doesn't have a true and defined nucleus, and also they are usually smaller. And for prokaryotic cell, they also do not have cell membrane. They do not have the membrane-bounded organelles. And here are the levels of the body organizations. So we as an organism, so inside our organism, we have various systems like the respiratory systems and also digestive system. Within the digestive system, we have the stomach and also liver and small intestine and large intestine. And inside that, it contains of a lot of tissues. And tissues is a group of cells that with similar functions. So here is the level of body organization. And finally, here is an extract from the HKEAA document. So you should be able to appreciate the technological development of the microscope, and also you should be able to prepare temporary mount specimens and identify different organelles as we discussed in this video. And you should be able to compare animal and plant cell and also prokaryotic and eukaryotic cell. So this will be the end of chapter 2. So see you next time.